Hello everyone, this is Dr. Yasmeen Idrees and I'm here for my second talk of the day. We counter a challenge which is urgent and deeply unequal survival gap in glioma patients in the developing part of the world when we compare it to the developed world. While the survival rate of glioma it continues to improve in the high-income countries, many patients in the developing part of the world they continue to struggle with a poor survival rate in glioma. Hence, I would like to present the topic Improving Survival Rate of Glioma in Developing Countries. I would like to thank Professor Patricia Tsai for supporting me for this talk. We have no conflict of interest to declare. A bit about my biography. I graduated from Fatima Jinnah Medical University, Lahore, Pakistan. Afterwards, I continued postgraduate training in general internal medicine and completed MRCP UK as a part of my postgraduate qualification. I find research quite interesting as it brings us all together. We can collaborate for learnings and can improvise healthcare for the betterment of the patients across the globe. Hence, I collaborate with Professor Patricia Tsai from Canada for the same. Background. Gliomas are the most common primary brain tumors originating from glial cells in the brain and spinal cord. Annual incidence of gliomas is around 7 in every 100,000 individuals. These tumors can be benign and malignant. Malignant gliomas, they form around 35 to 45% of all primary brain tumors. Amongst gliomas, glioblastomas are the most aggressive tumors associated with the poorest prognosis, and they account for around 60 to 70% of malignant gliomas, while their incidence increases with age. The glioma survival in global population-based data is poorest in glioblastomas. Median overall survival rate is 13 to 15 months. 5-year overall survival rate of around 4 to 17%. And these numbers, they vary in different settings and countries. Lower middle income countries, they have a poor outcome compared to high income countries. However, exact numbers, they are hard to interpret in the lower middle income countries due to limited data availability. Survival rate is not influenced by the biological nature of the disease alone, but factors like access to care, early detection and diagnostic accuracy, treatment modalities and follow-up for palliation and rehabilitation, they all contribute to a survival rate, which can be better for a patient if all these measures they are implemented timely. Objectives of this literature review included identification of gaps in the lower middle income countries by matching with the high income countries. Number 2, to highlight evidence-based strategies to improve survivorship of glioma cases in the developing world. Number 3, emphasize the significance of standard protocols for the overall management of glioma cases. Methods included a comprehensive literature review of over past 2 decades, where we gathered evidence-based practices which improved survival outcome in the developed world. Limited data availability, however, restricted access to real-time statistics and outcome in the developing world. Before I jump on to the main objectives of this talk, I would like to discuss briefly what are gliomas. These are the glial cell tumors, and the glial cells they form a supporting and nourishing network in the central nervous system. These cells are of different types, can be astrocytes, oligodendrocytes, or ependymal cells. Tumors originating from ependymal cells, they formulate ependymomas. From the oligodendrocytes lead to oligodendrogliomas. And astrocytes, they form astrocytomas or glioblastomas. Mixed tumors, they do exist as well. WHO revised glioma grading in 2021 based on their histopathological features alongside the molecular testing. And they graded these tumors from grade 1 to 4. The grade 4 tumors, they behave aggressively and they are associated with the poorest outcome. Grade 1, they are the less aggressive tumors and they show relatively better outcome. Glioblastomas are, however, almost always grade 4 tumors, therefore they are the aggressive forms of the tumors. Now, why there was a need for the molecular testing to be included in the grading system? This was because molecular testing is significant to identify the grade of the tumor initially. It serves as a diagnostic tool. But why is this diagnostic tool so important for grading of such tumors? This is because these markers, they serve for the prognosis and the prediction of response to the treatment, depending upon the presence or the absence of certain molecular markers. The IDH1 gene encodes for the enzyme isocitrate dehydrogenase 1 in the glial cells, and it is not mutated in the glioblastoma cases, whereas it is mutated in the other forms of the tumors. We all know that glioblastomas, they are the most aggressive forms of the tumor. Therefore, lack of mutation of the IDH1 gene is associated with a poor prognosis. And the tumors which have mutation in this gene, they are associated with a better outcome as they are less aggressive in their behavior. Now, the predictive nature of these markers is because of their response to the treatment modalities. One such marker is MGMT gene promoter, which is a DNA repair enzyme, and it gets methylated or not methylated by certain chemotherapies such as temozolomide. It leads to methylation of this promoter sequence. Those genes whose sequences they get methylated by chemotherapy, they show a better response to the treatment as they are responding to the treatment modality. Hence, the tumor will not keep on replicating at its own as its DNA repair enzyme is going to be killed. So, the molecular testing, it is very, very significant for grading to serve as a prognostic marker and a predictive marker for the glial cell tumors. Now, the important barriers to improved survival outcome in the developing world. The first and the foremost being community level barrier. Poor public awareness exists where the lack in the identification of the red flags or the severity of the symptoms. There is stigma associated with the brain tumors, and they can be late presentations due to poor socioeconomic status of the patients, impending early presentations or early identification of the tumors. Diagnostic challenges. Delayed diagnosis due to vague or misinterpreted symptoms is one. Poor infrastructure lacking CT scan or MRI scanning is another. Lack of immunohistochemical and the molecular testing is a third one. Treatment gaps do exist in the lower middle income countries. Shortage of trained neurosurgical and the neuro-oncological specialists. Existence of limited radiotherapy centers. High drug cost for the chemotherapeutics leads to their unavailability. And all these surgical resection of the tumor, chemoradiotherapy, they together form an evidence-based treatment modality which is known as STEP protocol for the glial cells tumors, and it has shown an improved survivalship in the high-income countries when practiced well and together. There are infrastructure gaps, poor referral systems, lack of integrated palliative and rehabilitation units, insufficient governance and poor policies, non-existent research and registries. They may hide actual numbers in such countries. How to bridge such gaps? First is to make sure that there is an early detection which can be brought in through public awareness, primary care training and referral systems, and utilization of telemedicine for screening and surveys for the symptomatic patients can serve for an early detection. Improvised management An affordable and adaptive protocol, which is relatively comparable to the high-income countries for the diagnosis and the treatment of gliomas in the low middle-income countries, can be very useful, including more national cancer centers where the deal with the gliomas too is necessary. Training the staff at critical level of care such as neurosurgical specialties is very, very essential. Defining care pathways is very important. Practice standard protocols for the diagnosis and the treatment to match evidence-based practices in the developed world, which shows improved survival outcome in glioma cases. Now, what are those evidence-based practices which led to an improved survivorship in glioma cases in the high-income countries? One is the early detection and diagnosis, and therefore CT scans and MRI scans, they are vital for every system. Molecular testing is another one, as it helps us diagnose, predict, and the prognosis and the response to the treatment of the tumor. STUB protocol, which comprises maximal safe resection, chemoradiotherapy of the glioma. It is an evidence-based strategy which led to an improved survival in the high-income countries. Targeted gene therapies, they are underway in the developed world. Palliation and rehabilitation systems, they are in place and they play a very, very vital role. Follow-ups, they are needed to help pick recurrence of the tumors. Now, the role of technology in bridging the gaps. Use of social media platforms to create public awareness. Use of portable scanners like MRI can solve the problem for scanning in the distant or the remote areas. Integrating AI tools for diagnostic interpretation can be helpful. Now, where is the role of governance? So, the role of governance comes into play to subsidize care and drugs, provision of generic drugs and radiotherapy, establishment of tumor registries, and funding to participate in global trials and establish local research networks to improvise healthcare modalities for glioma patients. Conclusion. Glioma survival gap is real in developing world, as the actual numbers may be much more than recorded due to limited data and lack of presentations and diagnosis. Hence, early and comprehensive management is necessary to bring an end to this solvable inequality in glioma survival. Key takeaways include: delayed diagnosis of glioma remains a major barrier to survival. Adaptive care plan through collaborative measures and training of the specialties is an unmet need of ours to save lives in glioma cases in lower middle income countries. I would like to acknowledge the support from members of International Oncology Cooperation Research Group for supporting me. Thank you.